length over here, right? So this is a 3 sequence length, this is a 5, this is a 4, 7 times. So for in a specific test tube, you would get a different amount of sequence length. And once you get that, once this cleavage process is basically done, then you go for your electrophoresis process, whereby you have these wells, and in these wells you would like load your samples, and then you run your electrophoresis sample. Once electrophoresis is done, you will get something of this sort, specific strands that you get at a specific sequences or nucleotide sequences. Now the main thing that we have to understand here is how do you actually interpret this result, right? So let's say you are getting a sequence band at 5 here in A plus G as well as G, right? So what this means is that when you are applying these this particular nucleotide, let's say only G cleavage, then you got a band here, but you also got a band at A plus G. Then what this specifically means is that G nucleotide is basically common for these two bands, right? And that's why you write G here. For example, in C plus T, when you applied, you got a band at C plus T. Now let's say you applied a specific band for C, but you C cleavage, but you don't got the band here, but you got the band for C plus T. Then that means that here only T is present. Okay, so different chemicals, what basically means is, just a second. Yeah, so different chemicals basically means you apply different kind of combination of chemicals that you apply. For example, let's say for guanine, you are basically applying a dimethyl sulfate, which is followed by a piperidine. So this chemical will specifically cleave at your G sequence. Let's say you apply dimethyl sulfate with pyropiperidine, then it will give you a pyrimidine basis, basically C and C plus T sequences. So in that order, you are basically going to try and apply different combination of chemicals, like permutation combination of chemicals, which will give you the final output basically, right? So piperidine is basically used for your guanines, and piperidine is basically used for your pyrimidines basically, right? So in that way, you are basically going to apply different combination of chemicals in order to get different bands that you want to get, or different cleavage sites that you want to get, right? Then what happens here is that once you get a band over here, then you see here we are getting two bands, right? So we can see that if you apply, let's say you applied only for G1 and you got a band here, but also you got the band for A plus G as well. So we know that in A plus G and G, G is common to these both, right? Because if it would have been A over here, then it wouldn't have been coincided with this G. It would have been upside, or a band up or a band down, but it wouldn't have been coincided with this one. Then we can say if these two bands are coinciding together, then obviously G is common to both. That's why the final sequence here would be G. For example, when you applied a G over here and A plus G over here, you didn't got the G band here, but you got the A plus G band over here, right? Then you can obviously say that in A plus G, you are getting A over here, because if it would have been G, then you would have got a band over here as well. But we are not getting a band over here. That means in A plus G, the sequence is A. So in that way, you apply the analysis process. So if two bands coincide with each other, A plus G and G, so the one which is common to both will be the sequence over here, right? And if these two bands are not coinciding with each other, then the uncommon nucleotide will be the sequence over here. The same goes for here, C and C plus T as well, because these two bands, like they overlay with the same number, then the common one will be the sequence. So C will be the sequence over here, right? And similarly here, you can say these are not coinciding with each other at the same number. That's why T will be here rather than your C, right? So in this way, you can write down your sequence, and then you go for, obviously, you will finally go for your radioautography, and then you would read your sequence, and then this will give you the specific sequence in which your DNA, single-stranded DNA is basically bound to happen, or the sequencing process occurs. So this was a very basic general overview of what maximum Gilbert sequences happen. Now the drawback with this particular sequence was that you can't apply all of these processes together. You can't mix all of these samples together, right? You can't, in a same test tube, you can't add A plus G and G as well, because then there's no point in bifurcating the process or bifurcating the results that you want to get, because then everything will be mixed up over here and you won't get the desired result that you want. So what happens is, because you are going specifically for a specific cleavage, it's a very time consuming process and it would require a large amount of time, a large amount of patience. So that was one of the major drawback that was there with the maximum Gilbert methods. And that's why the next process which came was for the Friedrich Sanger, or what is also known as the Sanger's method, right? Now, what happens in a Sanger's method is first we have to understand the basic structure of how your nucleotide basically looks like, right? So you have this nucleotide, and this nucleotide basically consists of three things. One is your nitrogenous base, which is followed by a pentose sugar, and then a phosphate group. So basically, it is a three phosphates attached together. So P, P, and P. So three phosphates will be together. Now, this is a basic structure of your nucleotide. Now, what happens is these nucleotide also have, let's say, 2' and 3' OH group. Now, if you remove this 2' OH group from your pentose sugar, if you delete the 2' OH group, what you get is this type of deoxynucleotide, right? So this is basically a nucleotide. Now that you have removed the 2' OH group, you are removing a hydroxy group. That's why the term deoxy, that is, you are removing the OH group. So that's why it is a D over here. And the step that is the structure that you are left with is known as your deoxynucleotide. And we also, in short term, we call it as DNTPs. And we know that deoxynucleotides are really important because for the DNA processing to happen or DNA sequencing to happen. Because DNA polymerase, when it starts the sequencing of your DNA molecule, it needs DNTPs to carry out the process or start the process or keep the process going on, right? Now, what Friedrich Sanger thought was what Friedrich Sanger realized was that whenever your reaction takes place, whenever your DNA sequencing starts or DNA polymerization starts, your DNA polymerase comes and then a new nucleotide will come out. So as soon as this new nucleotide comes in, the nucleotide which is already present in your DNA that has a 3' OH group extending out, right? So we have this 3' OH group which is freely available. And let's say a new nucleotide sequence comes in. So the lone pair on this oxygen can actually go and bind with the alpha phosphate group here. So this phosphate group basically consists of three phosphates, P1, P2, and P3. So this OH group can go and bind over here. And that's how the process, goes further out. Now, what Friedrich Sanger thought was that if this 3' OH group is not present over here, then obviously the chain will terminate, that the process will not go in the further direction because there is no freely available 3' OH group, right? So at that time, what happens is that what we are left is known as dideoxynucleotide. That means you have removed 3' OH as well as 2' OH. And as soon as your new nucleotide comes in, it doesn't have a free available 3' OH group with which it could bind and carry out the process, right? So the process in this Sanger's method is also the same. You first obtain your dsDNA. Using your heat, you denature your DNA. Then you make copies of the segment, that is a single-stranded sequences. Then you attach a short primer, which will actually initiate your process. Once your process is initiated, you add 4 polymerase solutions. So you have 4 test tubes. Let's say in this test tube, you add DATP, that is deoxyadenosine triphosphate. Then you add DTTP, that is deoxythymine triphosphate, then DCTP, and then DGTP, right? Now we know that if we only add deoxynucleotide, the process will happen because there is a freely available 3' OH group, which is available for a new incoming nucleotide to happen, right? Now, what Sanger did was he removed the 3' OH group as well, thereby creating a process whereby no freely available 3' OH group was left out. So when a new nucleotide comes in, it doesn't have the phosphate group of the new nucleotide doesn't have a 3' OH group to interact with, and thereby the chain terminates. So once you first add your DNTPs, and then the complementary strand will grow out accordingly because new nucleotides are coming in and you are adding DNTPs. So 3' OH groups are present. Now, as soon as you get a desired length, let's say, then what you do is you add DDNTPs as well. Now, once you add DDNTPs, then obviously what happens is as soon as your DDNTP go and integrate here, this chain is terminated because 3' OH group is removed from here and no further nucleotide can come and bind over here. Then what happens is you are just left with DNA sequences or DNA strands of varying length. And then you denature your grown chain, whatever chain you have observed, And then obviously the final step is you go for electrophoresis, you get the bands, and corresponding to that band, you get the sequences that you have. Corresponding to individual nucleotides, you will get a band over here, and then you can just write whatever sequences that you have. The main thing to note here is that the sequences are never read from top to bottom. The sequences are always read from bottom to top. So this will be your sequence: T, C, G, A, T, G, T, C, A, G, G. So this will be your sequence. This is one very important thing when you are going for a Sanger's method of DNA sequencing, right? So the takeaway from this particular process is that instead of using a 2' OH, you're also removing your 3' OH group as well. And that creates a process whereby no incoming nucleotides have the ability to go and bind to your 3' OH group, and that terminates the chain. That's why the name chain termination method. That is, you are basically manually you are restricting the chain so as to get your desired DNA sequence, right? Now, the Sanger process was really effective for a long time. But another major issue with Sanger's method was that the same thing happened here as well, because you're having only 4 test tubes, and in each test tube you can only add one type of DNTP. You can't add a mixture of DNTPs. Again, your process became slowed down, right? Because for each individual DNTP, you have to wait for this reaction to happen. So for the first reaction will happen in the first test tube, the second reaction will happen in second test tube, and so on and so forth. So you have to wait for quite a long amount of time in order for your process to run, and then you will go for your sequencing process or electrophoresis and then radioautography. So it was again a time consuming process, and people started figuring out that this is not the right way we should do it, because it is consuming a lot amount of time and we don't have this much time to spend. We want something new which could process out our result much faster. And that's when your NGS or next generation sequencing came into picture. So what next generation sequencing basically does, it is also called as your massively parallel or deep sequencing. The name massively parallel is basically from the terminology that what you're doing is you are parallelly running sequencing multiple times. So you're not running sequencing one strand at a time or one DNTP at a time, but you're running it multiple times parallelly. So what that gives you, it gives you a boost over all the other classical sequencing methods, thereby allowing you to process the results much faster as compared to the previous classical sequencing methods, right? So that is how you can go for your NGS. And then using an NGS, basically people have created your whole genome sequences based on that, and you can sequence large amount of huge set of data like megabytes and gigabytes of data. You can simulate those data, you can sequence those data in a really short amount of time. Now, when you go for a next generation sequencing process, there are various platforms which allow you to do those sequencing processes, right? So a very major company is called as the Illumina. So Illumina provides you a platform whereby you can actually go and do your next generation sequencing. So in order to do the next generation sequencing, you have to have first the general data set ready with you. So for example, let's say once you do an HPLC, so let's say you're doing an HPLC for a protein. So you get a curve, you get a graph, right? Whereby you can basically check how the purity of the protein is happening, right? Now, what happens is you have the luxury to store the data in any format you want, right? For example, you can store the data in a graphical format if you want. Let's say you don't want a graphical format of data. What you want is to store the data in an Excel format, so that in a later on, in a future direction, what you can do is you can utilize that Excel sheet of data and then create that graph again. So you can store the data in an Excel format as well. You can store the data in a text format as well. So it is your liberty via which you can store the data in any format that you want to. So before proceeding on with next generation sequencing, the same thing applies here over as well. You have to have your data set ready with you. You can upload your data set on a cloud server wherever you want. And then there are different platforms that provide you with the opportunity whereby you can actually upload your data sets and then do the sequence analysis. Let's say if you have a sequence data set of, let's say 1 GB or 5 GB or 10 GB of data sets, then running them on your normal desktop or a normal laptop won't be a very good reference point, because your laptop might crash down or your computer might crash down because of less amount of RAM that you have. So that's why people come up with data servers that which actually allows you a good computational ability or good computational process and speed, so that you can actually do the process much faster. And these are the different platforms which actually allow you to do perform NGS analysis, like Illumina, MISEC, Roche 454, Complete Genomics, Life Technologies, Ion Proton. So these are the different platforms whereby you can actually go out there, upload your data sets, do the simulations, do the sequencing, and then do the data analysis and get the final desired results, right? Now, what happens is the NGS method, basically it applies the basic methodology behind an NGS is four steps. So basically 4 steps are involved whenever you go for an NGS methodology. The very first step is the generation of libraries. Now, as we know, in any kind of a DNA sequencing, the very first step is the generation of libraries, followed by the sequencing, followed by your data analysis or the data generation. What next generation sequencing allows you is it allows you a new step over here, which actually optimizes your process. So the very first step is the generation of libraries. So you would generate double stranded DNA generation. Oh, sorry, it won't be P, it will be O here. I'm really sorry. So generation of DNA sequencing libraries by clonal amplification. So you isolate your DNA of interest, then you go for clonal amplification, whereby using PCR you can just create multiple large amount of DNA sequences that you want, and thereby you created a library, and that library has your DNA sequence of interest. So now that you have created a library, the next step and the possible step is to go for sequencing analysis. So what this basically does is, because now that you have your library ready with you, what is the need? Now, what do you want to do with that? Because if you don't want to do anything with your library, then there is no point in generating a library, right? So once you have generated a library, you go for your sequencing method. So the sequencing method, you DNA sequences by your synthesis method. So synthesis method is basically the method whereby you will apply your DNA polymerase, you will add a primer, and based on that, your DNA will be sequenced out. Now, what NGS provides you is a parallel sequencing as well. That means you're not just sticking with the classical method, you are actually segregating and amplifying the DNA at the same time while you are sequencing it as well. So you are sequencing the DNA, and at the same time, you are also running parallel sequencing. That means you are also segregating your DNA of interest, as well as when you are getting the DNA of interest, you are also amplifying that DNA of interest so as to create your final library or the final library of interest whereby your DNA sequences will be there. So this library was a general sort of library, we can say. That means you don't know what sequences were in this, right? So you have created a library, and once you have created a library, then you go for sequencing methods, and then after that you are parallelly running the sequencing methods whereby you are actually isolating your DNA of interest, and then you are also amplifying that DNA. And once you are done with the parallel sequencing, you go for data generation, and that data generation is a large amount of, it takes a huge amount of time to generate the data, because the data that you get after parallel sequencing is somewhere around 7 GBs or 10 GBs, depending upon the complexity of your DNA sequence. So if it is a much larger DNA sequence, then obviously it would take large amount of time for your data analysis to happen. For example, it would take, for example, a 7 GB data file could take you somewhere around 2 to 3 days, depending upon the server availability. So it will take around 2 to 3 days for your DNA analysis to happen, and then you would get your output thing that you want, right? So that is how your basically NGS is providing an edge over the others, is at step number 3, that it is providing you a parallel sequencing opportunity, a parallel sequencing platform, whereby simultaneously you are sequencing as well as segregating the DNA of interest as well and amplifying it as well, right? That is how you can actually use your NGS for your own preference or for your own advantage as well. And finally, you will get your DNA data generation. And once your data generation is happening, then you can just go for your final analysis using the online servers which are provided there, right? Now, the next thing is there are different kind of sequencing approaches which are there for NGS, right? Now that you're using NGS, what approaches should be the best, right? Because you have various kind of approaches. Because the basic approach is the same. You create a DNA library, you go for sequencing, and then you go for data analysis. But the second step, whereby you are basically using the sequencing approach, you can try and modify that process according to your need. People go for different methods according to their need, right? So the very first I will talk about the basic 3 methods that people go for. So in the second step, what you can do is you can go for a pyrosequencing method, the sequencing that you are basically doing. Now, what pyrosequencing basically is, the pyrosequencing methodology is basically based upon the idea of generating or detecting the light that you have. So what you basically do in pyrosequencing is that you have your DNA sequence ready with you. So once your DNA sequence is ready, then you go for you have your genomic DNA ready with you. Now your genomic DNA is a very complex structure, right? And you can't run that complex structure for your sequence analysis. So the very first thing you do is to fragmentize your genomic DNA. So you fragmentize your genomic DNA into small double-stranded DNA molecules. Now, once your small double-stranded DNA molecules are obtained, you then conjugate it with an adapter molecule. Now, the reason we are going for an adapter molecule is basically adapter molecule will allow this polymerase to identify your DNA of interest, and then you can go for your processing or DNA sequencing further down the road, right? Now, once you have your adapter attached with your double-stranded DNA sequence, then you cleave the double-stranded DNA sequence and prepare your single-stranded DNA sequence. Now, once your single-stranded DNA sequence is ready, you have a structure which is basically your single-stranded DNA sequence attached with an adapter molecule over here, right? Now, the next thing is, this pyrosequencing method is basically used in 2 ways. Either you can go for a solid pyrosequencing or you can go for a normal liquid pyrosequencing. Now, the reason, the basic difference between these 2 things is that in a solid pyrosequencing, when you are basically doing your process or experiment on a this one slide or a lab or a particular instrument you have over which you do it, it's a type of a well where you actually do the sequencing analysis. You don't want your DNA to be wasted out when you wash the sample. So in order for that, what you do is that you attach your DNA to a solid surface so that your DNA don't when once you wash the surface, your DNA shouldn't wash off, right? So what you do is that you attach your single stranded DNA molecule to a bead, and that bead attaches your DNA molecule. So the reason how that bead attaches is that, let's say this is a specific bead over here, right? A bead, let's say there's a bead over here. What happens is that in a bead there are different sequences which are attached, and in your single stranded DNA sequence there was an adapter molecule. So the sequences that are attached on this bead are complementary to the sequences which are there on the adapter molecule. So once your adapter molecule finds your complementary sequence here, it will attach to it, and then what happens is your DNA sequence of interest, the single stranded, it gets fixed. Now if you wash your like device or instrument, your DNA wouldn't wash off, right? Only those DNA would wash off that are not able to bind. So your bounded DNA won't wash off. Now once you are bounded with the DNA, then you can go for the next thing. So the next thing is, once you obtain that, now your DNA is fixed on the surface, then we transfer this bead plus our target DNA onto a well. It is basically for example, you have your microtiter plate, there is a well sort of thing. In the same way, you have same thing for an NGS as well, whereby you apply your samples on the well, and then once you apply the samples on the well, then you apply your new DNA polymerization, right? So you go for DNA polymerization, new nucleotides will be coming out. You have applied your DNA polymerase over here. There is a free 3' OH group available here. A new nucleotide will come. The alpha phosphate group of that nucleotide will get attached to this 3' OH group. And once that attaches itself, your rest of the 2 phosphate molecules is released in this form, and this is known as PPI or pyrophosphate. And that's why from here the name of your process came as pyrosequencing method. So you are releasing basically a pyrophosphate. Now what happens is, after your pyrophosphate molecule is released out, you apply what is known as APS, that is ammonium persulfate, and you catalyze this reaction with an enzyme known as sulfurylase. What this process basically does is that it converts your pyrophosphate into an ATP molecule. Now the reason why we are going for an ATP molecule is, initially we told that pyrosequencing is a method whereby you are actually detecting the light, right? So in order to detect the light, you are first going to create an ATP molecule. Once you have created an ATP molecule, then you apply another molecule called as luciferin. Now this luciferin molecule is basically found in the fireflies that you have, right? So you see fireflies in the night that they give out specific light, and that fluorescence is basically from this particular protein called as luciferin. So you apply this over here, and then you have ATP, and then you apply another enzyme called as luciferase. Now what this luciferase basically does is that it cleaves this luciferin, And as soon as this luciferin is cleaved, you get a fluorescent, a very large amount of fluorescence that you have. And that fluorescence is basically detected by a detector, and correspondingly you will get a light peak over here. And depending upon the peak that you're getting, you would be then finally considering the fact that your sequencing is actually happening or not. So let's say here it was your T, thymine, right? And then a new nucleotide came in. Let's say it was your DATP. So A will appear here, and A will get bounded to T. Then pyrophosphate will released out. This pyrophosphate will go and bind, convert into ATP. Then you apply luciferin, you apply luciferase. This luciferase cleaves luciferin, and then your fluorescence is released out, and you get the fluorescence in the detector. So you can say that now that I have now when I apply T over here, the corresponding sequence has been created because A was here. Let's say C came in. So C is not complementary with T, right? So this process won't be created. You won't get this specific light over here. And that's how you can tell the difference whether the sequencing is actually happening or not. Now you can update this process according to your need. You can actually do this in a solid state, or you can go for in a liquid state as well. And now in liquid state, what basically you do is, because in liquid state you can't wash off, right? Because as soon as you wash off your device, your target DNA will also wash off. Right, then you are not left with anything to go for with the experiment. So in a liquid sample, you don't go for wash off. What you do is you apply another enzyme known as epirase. What this epirase basically does, it does the same process over here, but without actually making your DNA stick to a solid surface. That is the only difference between a solid pyrosequencing and a liquid pyrosequencing. And the final basic idea is that you are released with a pyrophosphate molecule. Then this pyrophosphate along with ammonium persulfate and sulfurylase is converted into an ATP. Then you apply luciferin, you apply luciferase. This luciferase cleaves luciferin, you get your desired immunofluorescence, and this fluorescence is then detected in your detector. And based upon the peak, you will tell how much of your DNA sequencing is happening and whether the strength, what is the strength of your DNA sequencing that is basically occurring. So this is the basic overview of how pyrosequencing happens, right? Another approach for your NGS, what people go for, is sequencing by reversible terminator chemistry. What happens here is basically you are having a flow cell here, and then these are basically the short primers which have a specific sequence along with that. Then obviously you have your double-stranded DNA. You convert that double-stranded DNA into a single-stranded DNA, and then you convert that single-stranded DNA, and then you apply that DNA over your flow cell. Now, as soon as you apply the DNA over the flow cell, now this DNA, if it finds a complementary sequence, it will go and bind over here. Now, what happens here is that this portion becomes rigid, right? But this portion is still flexible because this particular portion hasn't bounded to anything. So what happens is this particular portion bends over, and then this region of the DNA, the terminal end, it finds another complementary sequence if it is present in the flow cell or not. So if it finds another complementary sequence, it would bend down and go and attach to the complementary sequence over here. Now, as soon as your DNA bends over and then it forms a bridge-like shape and it finds the complementary DNA sequence, the next step that you go for is you apply a primer. Now, the reason that you are applying a primer is that you are basically going to convert this ssDNA into a double-stranded or a complementary strand. So you are going to basically create a complementary strand of this single-stranded DNA. So this primer would create a complementary strand over here. And as soon as a complementary strand is created, this particular DNA molecule, the double-stranded that you have created, this will then cleave off from here. And the reason how it cleaves off is you apply another enzyme that basically cleaves this particular DNA sequence from here. So now you initiated with a one DNA sequence, but now you have two DNA sequences over here. So one is a forward strand, another is a reverse strand that you created using your primer over here, right? Now this is just a short form, but actually when you apply this in an experimental procedure, you do it in a very huge amount of, like a very large volume of thing, right? So you get a multiple sort of things in this way. So you get clonal copies of your DNA sequences, and then obviously you can go for your different identification or analysis method. So let's say you are going for you can tag this, you can radio label your DNA sequence, and then correspondingly you will get your illuminescence, and then you can detect your illuminescence, which will basically tell you whether your DNA sequencing happened or not. So this is basically a reverse type of thing, because we are creating a reverse primer over here. That's why the process is known as a reverse terminator chemistry. So this is another optimization process that you can go for while you are doing a next generation sequencing. Another method that people go for And the final method that usually people go for is by sequencing by ligation mediated process, and they usually use the ligase enzyme. Now we know that in a sequencing process, DNA polymerase is involved, right? But what happens here is you are not using DNA polymerase at all. You are just using your ligase, DNA ligase. So first you have created your library, you have digested your DNA, you have created your single-stranded DNA with an adapter molecule, you have attached your single-stranded DNA on your magnetic beads in order for your single-stranded DNA to be fixed. You don't want your DNA to get washed off once you are washing your equipments, and then you go for PCR in order to multiply or amplify your sequences. And this is basically a glass slide whereby your DNA sequences will be stored out. So this is your general, how your general sequence of DNA would look like. So at the 5' end, you have this magnetic bead over here with a probe, radio labeled probe, then an adapter molecule that you have attached over here, and then the template sequence that you have generated for your DNA sequence. Now once this is done, the next process is that you apply ligation. You basically apply a ligase. Now what ligase basically does is that here your adapter and your template sequence are not at all joined together. There is a nick between your adapter and your template sequence. So in order to join that nick, what you do is you apply your ligation method, or you apply your DNA ligase. So once DNA ligase acts, what it will do is that it will join your adapter sequence with your template sequence. Now as soon as your adapter and template sequence are joined, after that you wash off your equipment. So whatever DNA sequences which were not able to get ligated, those DNA sequences are washed off, and those DNA sequences which were able to get ligated, those remain as it is, right? Then you go for your next step, that is you specifically, like you do for the probe ligation method, you do the fluorescent labeling. So whatever bases that you are basically adding, those are radio labeled. So as soon as your base gets attached to a new nucleotide, your fluorescence will be there. You detect the fluorescence, and based on that fluorescence, you will then detect what is the final output or the sequencing that is basically happening, right? So here what happens is, first you go for a universal primer hybridization. So you have a universal primer that you are going to apply everywhere. Then you apply your tagged DNA bases. Then once the tagged DNA bases are here, fluorescence will happen because the pyrophosphate molecule will release out PPI, and then corresponding to that, the radio labeled fluorescence tag will also get quenched, and that fluorescence will be detected. Then you cleave off the fluorescence, and you repeat the same step over and over again until you get your desired result. Let's say once you get your desired result, then you go for your probe reset. That means what you can do is that once you are finally like happy with the sequence, that is okay, this is the length that I want. Now I don't want to carry out this process any further. So you can manually restrict the process. You can just remove the primer from the sample, and your process will be halted at that particular point of time.